FADIC offers a variety of international online trainings accredited by FADIC and provided by well-reputed professionals in the field, which means that you can always keep up with the latest topics in your specialty wherever you are and whenever you need it.
بسم الله الرحمن الرحيم. الحمد لله والصلاة والسلام على رسول الله. أخواني الأعزاء، أخواتي، أسعد الله أوقاتكم بكل خير. معكم أخوكم سالم مجري، معيد بجامعة الملك خالد. حاليا أحضر الماجستير في جامعة إمبريال، ويشرفني أن أكون أحد أعضاء تطوير مهنة الصيدلة. أحب أشكر في البداية كل من ساهم وشارك في تنظيم هذه المحاضرة، وأخص بالشكر أخي فارس الرشيدي. الشيء الثاني، أرحب بالأعضاء اللي خارج الجروب، أقصد جروب تطوير مهنة الصيدلة، ويعني أستغل الفرصة هذه إني أدعوهم، خصوصا أغلبهم صيادلة، إنهم ينضمون للتجمع هذا، وممكن يتواصلون مع الأخ فارس. طيب، بسم الله. Pharmacogenomics and personalized medicine. Basically, the main goal of healthcare professionals is to provide a better diagnosis, easier intervention, and more efficient drug therapies. And these can be achieved by using personalized medicine. Personalized medicine is also known as individualized medicine. Pharmacogenomics play a central role as a tool of personalized medicine. And today, or tonight, we are going to focus more on pharmacogenomics. Before running through the lectures, I would like to highlight the main objectives of this lecture. So at the end of this lecture, I hope everyone should be able to define key terms such as pharmacogenomics, pharmacokinetics, pharmacodynamics, and so on, understand the basic molecular concepts of pharmacogenomics, and identify genetic variations such as mutation, single nucleotide polymorphism, and so on, and give an example of clinical application of pharmacogenomics. Pharmacogenomics or pharmacogenetics. To be honest, there is no main difference. I have asked many professors here at Imperial, and they said there is no difference at all. But I've quoted the definition from FDA for pharmacogenomics, which is the study of variations of the DNA or RNA as a resulted to drug response. And pharmacogenetics is the study of variations in DNA. So just pharmacogenetics mainly for DNA, and pharmacogenomics is more broad and include RNA and DNA. So this is the main difference. And both terms are used interchangeably, I mean. So don't confuse about this. And of course, everyone can define pharmacokinetics and pharmacodynamics. And SNP, pronounced as SNP, which is a single nucleotide polymorphism. We will discuss this later. Last two things is genotype and phenotype. So genotype is genetic information contained within the DNA. So the sequence of DNA is called genotype. And phenotype is the physical results of gene expressions. So what is the sequence can include this result as phenotype. So phenotype is the result of genotype. Here I would like to make a comparison between traditional prescription of drugs and the prescription based on pharmacogenomics. To be honest, if I if I have, for example, same patients with the same diagnosis and same variation factors such as gender, age, and all of them are healthy, and if they have the same diagnosis, I can start using initial doses. And this is the result. If we start with initial doses, we will find different response to drugs. So most patients has effective doses, okay? But we have some groups develop different responses. And the reason for that is variation in the genetics. And we will discuss that in coming slides. But here, what happens is in this patient, they develop toxic dose because they break down drug very slowly. So what is the solution of that? You can decrease the dose to achieve desirable effects. And here, these patients, has no effect, and the reason of that because they break down drugs very quickly, so we need to increase the doses to achieve desirable effects. And the last one has no effect at all because we choose wrong drug. And the reason for that because there is no receptors in these patients. So we have to choose another drug. So the main goal of pharmacogenomics is to have right dose, right drugs with right dose to right patient. And you can see here US FDA approved drugs that contain pharmacogenomics information in their package insert labels. And you can see here different drug classes that mainly based on pharmacogenomics. As I said, the main goal is to enhance efficacy and decrease toxicity of drug. And why we need that? Because as you can see here in our chart, the most efficacious drug is analgesic, but they can achieve up to 80% in terms of efficacy. And most drugs range between 40 to 60% in terms of efficacy. So we need to enhance efficacy more. And this can be achieved by using pharmacogenomics. On the other hand, we need to decrease side effects of drugs. And if you can see, this is few facts about the adverse effect of drugs. So in the United States, more than 100,000 deaths annually. And the cause is side effect of drugs. And it considered the fifth cause of death. And nearly 70% of hospital admission is due to adverse effects. And the cost of adverse effect is approximately $136 billion yearly. So based on these facts, we need to work out more, to investigate more, to decrease the toxicity of drugs. And as I said, it can be achieved by using pharmacogenomic tests and information. I would like to start before mentioning the application of pharmacogenomics. I would like to start with molecular basis of pharmacogenomics, because it's important to understand why there are huge variation between individuals. Basically, in the chromosome, there is double-strand DNA. And if you can see inside these genes, there is single nucleotide. So single nucleotide composed of purine, which is adenine and guanosine, and pyrimidines, which is thymine and cytosine. So if there is a change only in one nucleotide, it's called single nucleotide polymorphism. And this change will change the cascade of production. So if this gene, for example, includes protein, this protein may have different function. This, for example, if this gene includes enzyme, this enzyme could be more potent or less potent or inactive. And the human body contains about 20,000 to 40,000 genes, include variations based on this statements. And as I said, a variation at a single position in the DNA called single nucleotide polymorphism, and it accounts for 90% of a human genetic variation. Also, this is an important slide because it helps everyone to understand the nomenclature of genes. So there are two types, common two types of nomenclature of genes. So first one starts with genes and number and two letters. So the gene, the first letters is the name of particular gene. And the numbers here represent the position of nucleotide. And first letter represents the original nucleotide, and the second letter represents variant nucleotide. And here the first letters represent the name of particular gene, and the star and the number after star represent the allelic variant. And if you have two stars, that means they represent two allelic variants. Now we want to see the effects of molecular changes and the application of these changes in practice. Nearly about 75% of people have genetic variability that influences drug response by affecting three things. First, drug metabolizing enzymes. Second, drug transporter proteins. And the third is drug receptors. And these can be divided into pharmacokinetic variability and pharmacodynamic variability. So let's start with pharmacokinetic variability. So pharmacokinetic is the effect of the body on drugs, which means absorption, distribution, metabolism, excretion. But in this lecture, we are going to focus on metabolism. So metabolism can be divided into phase 1, which is mediated by cytochrome P450, and phase 2, which is conjugations. So let's start with pharmacogenomics of cytochrome P450. People can be divided into 3 types: ultra metabolizer or extensive metabolizers, poor metabolizers, and ultra extensive or rapid metabolizers. And these categories based on how they can metabolize drugs. And also, I'm going to mention just the 3 types of cytochrome P450 because they are the most extensively studied subfamilies. So the first one is cytochrome P2D6. This metabolizes 30% of the drugs. And this is the common classes: antidepressants, beta blockers, and analgesics. So when I mentioned these classes, it doesn't mean all drugs belong to classes, but some of them or most of them. And also it doesn't mean all drugs in this class mediated by cytochrome P2D6. It could be this enzyme or another enzyme or more than 2 enzymes. But we have here 3 major different allelic variations. So as I said, this is the name of gene, and the number after star represents the allelic variation. So based on pharmacogenomic tests, if we have this variation, the production of this gene is inactive enzyme. And this can be found on Caucasian people up to 25% in terms of frequency. The next one is cytochrome P2C1*5. As I said, this is the number of allelic variation, and this produces no enzyme, which means they are very, very poor metabolizer, and the frequency of this allelic gene is 5% in general people. And the last one, cytochrome P2C1*17. This includes enzyme with less activity, and its frequency is 30% in African people. So if we know this information, we can give better doses and therefore enhance efficacy and decrease and minimize side effects. And fortunately, genotype test of cytochrome P2D6 was approved by FDA in 2005. And this is the tool. It's very small and called AmpliChip cytochrome P450 array. and it's used clinically to determine variant genotypes. The second one is cytochrome P2C9. It metabolizes 15% of drugs, such as non-steroidal anti-inflammatory, angiotensin receptor blockers, and anticoagulants. So I would like to mention this example, which is warfarin. Warfarin is the most widely prescribed anticoagulant, and it's used to decrease the risk of blood clots forming. So basically, warfarin acts by inhibiting vitamin K epoxidase reductase enzyme, and this enzyme is essential for activation of vitamin K. And as you know, vitamin K is essential to or responsible for converting inactive clotting factors to active clotting factors. So warfarin is metabolized by cytochrome P2C9, and this can determine the dose and therefore the efficacy and side effects. As you can see in this table, warfarin pharmacogenomics is not effective. And if we start, for example, by 5 milligrams in those patients who have this allele, that means the development of adverse effects will be high. So based on pharmacogenomic tests, we can choose right dose for right patient. The last one is cytochrome P2C19. And this metabolizes several important drug classes, such as proton pump inhibitors, anticonvulsants, and antidepressants. Basically, there are major allelic variations can be occur in this enzyme. 2 of them can produce inactive enzyme or less active enzyme, and 1 of them produce very active enzyme. And we have to be careful with this one and with other one. And here is the example of drugs that metabolized by cytochrome P2C19. Clopidogrel is a prodrug, which means it needs enzyme to convert inactive form to active form for metabolites. So, based on this information, if we know patient has this allelic variation or this variation, we know this patient is at high risk of developing bleeding. Why? Because this drug is very, very active, and it will convert prodrug to very active drug very rapidly. So we should decrease the dose. And this is the opposite, which represents the first type. So here we can increase the dose to achieve desirable effect, because the converting of prodrug to active form is very slow, so we need to increase the dose to achieve desirable effect. And here we have to be careful with ultra extensive metabolizers, because they are at high risk of bleeding. And here poor metabolizer have a reduced antiplatelet effect, so we can increase the doses here. And again, fortunately, this type of cytochrome P has ampli chip as well, and it can be used clinically to detect and to determine the different variation in these genes. So now we will move to pharmacodynamic variability. As you know, interaction between drugs and specific targets in the body is the definition of pharmacodynamics. So any variation in these targets lead to a variation in drug response. And pharmacodynamic variability can be divided into variation in germline DNA or variation in somatic DNA. So, the variation in germline DNA, which is inherited mutations that arises from the germ cells, sperm or eggs. So, the genes that you got from your mother and your father called germline DNA. And here, two classic examples with beta-adrenergic receptors. So, as you know, beta-adrenergic receptors is main targeted by several drugs to treat many diseases. But in this case, patients homozygous for arginine mutations do not respond to beta agonists, particularly albuterol. Why? Because the gene that encodes beta receptors has variation or mutation. So this mutation change a little bit the conformation of receptors, and this change lead to deactivation of binding sites of albuterol. So albuterol has no affinity and no effect on these receptors. So if we use albuterol to treat symptoms of asthma, we cannot achieve any response in those patients. The second example is xylitol. Xylitol is inhibitors of 5-lipoxygenase enzyme, and also it's used to treat symptoms of asthma.